Diolch am ymuno ni yng nghanadledd Cenedlaethol Seren 2018. Ewch yn bellach. Thank you for joining us in the Seren National Conference 2018, Above and Beyond. Dyma sesiwn. This is session Gwyddorau Bywyd Meddygol, Life, Medical Sciences.
All right, so good morning and welcome. This morning we are going to be talking about how we can cure our own cancer, so how we can use things that the body produces in order to treat and combat cancer. Okay. So before we get on to any of the technical science details, I just wanted to briefly talk to you about how to take good notes. Normally this slide will prompt you to take out your notebooks if you haven't done so already. So you're always going to be told, take good notes, make sure you're taking notes, take good notes. Well, how do you actually take good notes that you're going to be able to use later on, okay? Often I find that I write pages and scribbles and this and that, and then I can't decipher it, let alone use it afterwards, and I kind of sit there feeling like it was a little bit pointless, me actually writing it down. So how can we take clear and concise notes that we can actually use later on, and how can we use them? So first of all, why? Why do we want to take good notes? It can keep you motivated throughout the session. If perhaps it gets a little tiresome in the last 15 minutes we've all been there, taking good notes ensures that you're still listening and helps to keep you a bit motivated along through the session. It requires you to think about what you're writing. I always find that if I physically write something down, it tends to cement into my brain a little bit more, or if I type something out, or if I say something, I repeat, something to somebody else. It sticks in my mind a little bit more than if I just hear it and try to remember it. Okay. It helps you to make connections between topics, and there's a method that I'm going to mention briefly in a moment that will help you to make those connections between your notes and help to keep them more tidy, easy to decipher, and less messy and all over the place. And of course you have this review material for after the lecture. It means you don't have to remember anything, you can write things down. Okay. And in university, they're usually delivered, the lectures are usually delivered in the format. They will have kind of bare kind of slides, and you'll be expected to take notes, or perhaps even fill in the gaps. They might miss out, the 3 ways to take good notes, and then they'll just have those bullet points, and they'll say them, and then you'll be expected to kind of fill them in, to kind of ease you into good note taking. So a really good way that I like to take notes is to divide my paper into 3. So you would have the margin as one small section, and you'd have a little strip at the bottom, another smaller section. The main large section would be where you take the main body of your notes. And then at the end, towards the last kind of 5 minutes or so, you'll then start to kind of condense and consolidate what you've been learning about into a couple of sentences, just so that you know the general idea of what you've been learning in that particular session. Then after the session, or perhaps towards the end if it's a particularly tiresome session, in the margin you'll then write some keywords. Okay, so for example today you could write good notes. You might write medicine, you might write life sciences, you might write cancer. You'll write kind of little headings like that, and that will help you to make connections between the notes you've just taken and previous notes, because you can connect them and they're easy to see, one words, and you can see if there are any crossovers between the notes that you're taking over the weeks or months. Okay. Other people like to use this mind mapping method. If you're a visual learner and you don't like to write down reams and reams of stuff, this can be a really nice method. You start with a general concept in the middle, for example, cancer research, and then you might branch out like a spider diagram, diagnosis, and then have some more going off there, treatment, and some more going off there, symptoms, causes, and you can start to build out. And again, it's nice and easy to make connections there because they're kind of one word kind of notes, and they're nice and easy to compare with others. Okay, and the idea is that you'll be building up, as you go through your academic life, you'll build up kind of like quite a large book of these notes, but you want to be able to use them. Okay, so these keywords, getting a nice fancy highlighter is always nice and nerdy. You can, do a bit of color coding. We all like a bit of that. So I'm going to give you a little overview of what we're going to do this morning. So we are a touch shorter on time than we would be normally, so I'm going to be getting you out of here in about 35 minutes, so that you've got time to look around the exhibitions and grab something to eat or drink before your next session. But in that 35 minutes, we're going to try and have a good understanding of what studying at university is like, okay? What opportunities exist. We know what universities exist. What opportunities exist for you to take advantage of, and how you can take advantage of those opportunities, how you can make the most of it, okay? In terms of the scientific content, we're going to have a look at the key questions we ask within life sciences and medicine, in particular cancer research, and we're going to have a little look at a case study of academic research, and that will be my personal research that I'm working on at Swansea right now. And finally, we're going to take a look at ideas and strategies that can be applied to studies that are going on at the moment. Okay, so I'm going to keep this fairly brief, but this is just me sharing with you my academic journey, just to try and hit home that it is not a linear path, okay? You guys probably had stress choosing your GCSEs, and then you have stress choosing your A-levels, you know, which ones are the ones that you want to do, which ones are going to lead you on the path, you know, the correct path. You know, now you're going to be thinking about what undergraduate degree do I want to do. Now there's even more choice, and it all starts to get a little bit overwhelming. But what I wanted to share with you was that my path wasn't exactly linear, and if you don't know what you want to do right now, that's okay, because often your bachelor's degree will be in somewhat the right hemisphere of what you would like to be doing, but it may not be exactly right, and you should just choose something that you're good at and that you enjoy, okay? So I, in my wisdom, opted for forensic science. It was just as cool as it sounded. We got pathology, toxicology, crime scene photography, all that kind of stuff, as well as the basic science. So that basic science, out of all of the cool stuff, was actually what interested me the most. So it helped to push me on to my master's degree, which I did in human and molecular genetics, which pushed me a little bit more towards the clinical pathway and the medical pathway, kind of leaving forensic science and CSIs behind. As I was no longer 18 by that point, it wasn't quite as cool anymore. So after my master's degree, I then panicked. You know, I need a job. You know, I need to earn some money. I need to actually go out into the real world for the next chapter of my life. So after applying for a while, I was offered a job in Cambridge, working for an antibody discovery company. Antibody discovery will make a little bit more sense when we get on to talking about my research, but very briefly, antibodies are the type of therapy that I'm working on for cancer, and in order to treat effectively, we need to use the right antibody. So we know that antibodies bind specifically to different things. One antibody will bind to one thing, but it won't bind to another. So we need to discover the correct antibody for the job that we want, okay? And that was what the company I worked for did. And finally now I'm at Swansea University studying my PhD in medicine, where once I've done a lot of reading and writing and experimenting, I'll be awarded my doctorate. This isn't to be confused with an MD. I will not be a medical doctor. I won't be a GP. So if you have any health issues, I wouldn't be able to help there. If someone calls for a doctor on the plane, that's definitely not going to be me. But I will still be awarded the title of doctor. And this is the highest kind of academic qualification that you could aspire for. So, you know, if a PhD is something you think in the future that you would like to go for, you don't necessarily have to do a master's in the middle. You can go straight from undergraduate straight to PhD. If anyone has any questions about that kind of pathway, feel free to come and find me after the session. So what does a typical timetable look like? This is a really common question. What you really want to know here is how much contact time am I actually expected to have to attend, and how much free time am I going to have left, frankly, to sit on my butt and do very little. That's the basic question there. And the answer is you will get gradually more and more free time as you progress through to your third year. So you'll have more of this contact time, so lectures and times where you have to be somewhere. You'll have more of that at the beginning in your first year whilst everybody's getting onto the same page, and you'll gradually have less and less and more independent study, not sitting on your butt, more and more independent study as you get towards your third year, okay? So you will be less busy than you are now. I imagine your schedules are pretty jam-packed, probably starting at 9 and probably finishing around 4 in the day. So that's already a very packed schedule. So you'll be having a lot more independent study at university than what you're used to right now. And I'm saying independent study, and I mean independent study. I don't mean a free where you pop off into town and get a pizza. I mean actually independent study. So just on that note, there were 2 pieces of advice that I received that I wish I'd have taken advantage of, but I didn't because I was 18 and I really did want to go into town or sit on my bar and eat pizza. But really, you should be using those personal study times to actually study. I know it's not a popular piece of advice, and it's very difficult, especially in your first year. You're away from home, you're living in a new place, you're living in a new city, you want to go out and do exciting things. But really, try and fit in your studies into there somewhere. It'll help you no end later on. It'll make things a lot less stressful, a lot more relaxing later on. And the second piece of advice is to read. Read things you like, read things that you don't like. Decide that you don't like them and read something else, and then read something else again. Okay, keep expanding your knowledge base, because you're only going to decide where you want to go if you read about it. Okay, and watching TV doesn't count. I'm sorry, but it doesn't. Okay. So now we are actually going to move on to a little bit of the science. We've got half an hour to get through the scientific content, okay? So we're going to have a little look at cancer research to start off with as a whole in general. So we would define cancer as the uncontrolled division of cells within the body. So cancer cells in particular are what we call proliferative. They proliferate heavily, they replicate more, they replicate faster, and they also evade this process called apoptosis, this programmed cell death, this very normal process in the cell cycle. Cells are made and they die. That is the way that things work in the body to keep things healthy. Cancer cells take no notice of this, and they do not die when they should, meaning that they build up, okay, they replicate even more, and obviously they have a mutation. That's what makes them cancerous to begin with, and you end up with a large mass wherever it may be, okay? So current diagnosis methods for cancers in generally include clinical imaging. So you might have ultrasounds or other types of body scans. You might have blood tests looking for certain proteins that are higher in your blood if you are afflicted by a certain type of cancer. Physical exams, if it's the tumor is somewhere where you can see it and actually physically examine it, then you will have that done. Obviously, that's not always the case. If you have, for example, brain cancer or lung cancer, you can't exactly get your hands in there and have a look at what's going on. And finally, the only confirmatory way to tell whether you definitely have a certain type of cancer is to actually take a piece of that tissue in what we call a biopsy. And this can be particularly stressful and unpleasant if your tumor is located in a place that's very internal. So, for example, in the brain or in the lung or in the reproductive system, there are definitely areas of the body where it is very difficult to get to, and this can be a really nasty process. So we're going to touch a little bit more on how we can improve these methods in order to get cancer diagnosed earlier and in a less stressful manner. Okay, the current treatment methods, 9 times out of 10 will be surgery if it's appropriate. If you can see and you can get to the mass, remove it. And we normally follow that up with chemotherapy or radiotherapy to clear up any cells that have been left behind that we haven't managed to catch and remove, okay? But it's really important to mention that the specific diagnosis method and the treatment pathway will be very specific to the type of cancer that we're dealing with. Just as extreme examples, leukemia is a cancer of the blood. There is no tumor, there is no particular site of infection, whereas, for example, breast cancer, there is an actual tumor there that you can see and feel and remove, meaning that they would have very different diagnosis and treatment methods. Okay? So we're going to have a very brief activity. We're just going to have 1 minute just discussing what questions do you think we are trying to answer in cancer research. It's a very big topic. There's a lot of groups studying many different things in cancer research. What do you think are some of the general questions that we're trying to answer in cancer research? So chat amongst your groups, and in 1 minute we'll come back together and I will ask some of you, if you're feeling okay, to put your hands up and share with the group the kind of questions that you think we're trying to answer in cancer research. research. So 1 minute, go ahead. All right, let's bring it back together. Does anyone feel brave enough to put their hand up? Oh, come on, it's not that scary. Hey, there we go. Yeah, absolutely. Prevention methods. Once we understand what causes a particular cancer, prevention methods are absolutely paramount. Prevention is always the best course of action. Fantastic. Yeah, so looking at preventative methods. Anyone else? Oh, you're all quiet today. Oh, yes. Absolutely. How can we replicate cancer outside of the body so that we can do our experiments, we can look at perhaps what's caused it, and we can test some of our treatments on it? Fantastic. Okay, so here are a few of the questions that I jotted down. So the big one that I put at the top is a little something I'm looking into, is talking about differences in our DNA and how does that relate to cancer, okay? So unless anyone is an identical twin in this room, we all have completely unique DNA within our systems. We may have similar genes. For example, we all have the gene for hair. We all have hair, but we will all have slightly different genes. Some of us will have wavy hair. Some of us will have brown hair. Some of us will have blonde hair. We will all have a very unique, completely unique set of genes. Now, how does this shape our cancer treatment? And how can it shape our diagnosis? We know that there are certain genes that are available at the moment that if they are mutated, then they increase our risk of cancer. For example, BRCA1 and BRCA2 genes, they are DNA repair genes. They focus on literally if DNA is under attack all the time, breakages do happen. The body naturally responds to this in a very normal way by recruiting these proteins that are produced by the genes BRCA1 and BRCA2, and they will repair any DNA breakages. If these genes are mutated and they aren't functioning properly, the repair won't happen, mutations occur, these mutations grow and divide, and we end up with potentially a cancerous mass. So, could it be that if we all in this room had the same disease, that we would receive a different treatment for the same disease based on our individual genetics, okay? To make the treatment more appropriate for us, less side effects. You know, perhaps somebody wouldn't need something as strong as somebody else, something that is the best for your body, okay? Can we reduce cancers to a set of common traits, okay? Can we advance on cancer as a whole by finding something that's common to all cancers, okay? Why should we care about the tumor microenvironment? Now, the tumor microenvironment is literally, as it says, is the environment that surrounds the tumor. So we're talking about things like the tumor will go through a process called angiogenesis, which is where it develops its own blood circulation system, which means that it can have its own oxygen supply and start to sustain itself and develop in that fashion. How can we target anything there? Can we use anything there to help to combat and treat the tumor? Does epigenetics play a role in cancer? So epigenetics is whether genes are turned on or turned off, whether they are expressing a protein or not expressing a protein. Does this play a role in cancer? Is there a particular gene that when turned off increases your risk of getting cancer? And how can we take advantage of that information that we've found out? Is immunotherapy a turning point in the fight against cancer? Immunotherapy, using antibodies as a targeted, direct therapy against a specific cancer. I'm a little biased, as you probably have gathered. This is my area of research. So yes, I think it is a turning point, but it's a very hot topic at the moment. Will there be any P53 breakthroughs? Now, P53 is a very, very common gene that's mutated in cancer. It is a tumor suppressor gene, and we all have it in our bodies right now. Right now, it's suppressing everybody from getting tumors. That's its main job. It regulates cells growing and dying. It makes sure that this is happening in a controlled and normal manner. When this is mutated, the cells will grow in an uncontrolled fashion, and that's when we start to get masses. So P53 is mutated in many, many, many cancers, and it is coming very close to one of those common traits among all cancers. Are there going to be any more breakthroughs here, or is it time to move on to another gene to see if any of the others have got any kind of therapeutic potential over here? Can we exploit the differences in cancer cell metabolism? So metabolism being the biological and chemical processes that go on in order for the cell to survive and maintain a normal life. We know that cancer cells divide more readily than they should. We know that they evade dying when they should. Can we take advantage of this knowledge in order to treat cancer? And finally, are mouse and animal models still relevant, or are there other models, as we spoke about over here briefly, are there other, for example, 3D models that we can, we now have 3D models of the lungs, the heart, the reproductive system. Can we model organisms so that we don't have to use animal models, and are they more appropriate to be using? So we can narrow these down into a few more general questions. Is personalized medicine possible, where each of us would receive a different treatment, even if we had the same disease? Will there be one cure for cancer? Can we find that common trait between all cancers and cure it once and for all? What is the best model to study cancer? If it's not rats or mice anymore, what is it? What is the most appropriate method that we have to study cancer at the moment? And finally, what biological factors can affect cancer diagnosis, prognosis, and treatment? Prognosis being the pathway in which the disease that you have is going to take. If you have a good prognosis, then it's likely that you will recover from your disease and you will go on to lead, a completely normal life. If you have a poor prognosis, it's likely that you will continue to suffer, or suffer even worse from your disease, and potentially even succumb to your disease at some point in the future. So how do we try and answer these questions? There are a lot of them here. We like to set specific research questions. We like to divide off into groups. So for example, my group is looking at gynecological cancers. Another group will be looking at brain cancers. There will be blood cancers. We split off into little groups and we set very specific questions, much more narrowed down questions than we were thinking about in the questions we did for our activity. So we also do a literature review, which is we basically survey the current literature that's available about our chosen topic, what is going on with it right now, and how can we figure out how to build upon that. We obviously like to do experiments in the lab. We test out our treatments. We figure out how things work. It's a big part of it. We like to publish papers as academics. We like to contribute to that literature that's available. That's how we like to disseminate our knowledge between other like-minded individuals. And of course, we love to present at conferences. We like to present at academic conferences for students to get involved, to share our knowledge, to see if we can recruit more people that are interested in this field. And we also present at more academic conferences to a slightly more scary audience full of university professors that know exactly what I'm talking about and more, and we can help each other out there by asking each other questions and using each other's knowledge to make the best progress possible. So some of the debates and disagreements we have within cancer research, a big one is how have we validated our technique? How do we know that what we're claiming to happen is actually happening? It's usually a very small process that we're looking at on the cellular or molecular level. How do we know that these things are actually happening? Does your hypothesis hold true in human models? There's a lot of research and data that we have to have behind us before we move on to these clinical trials into human models. So really, we don't know until we get there. Some treatment or target can look incredibly promising until we put it into clinical trials, and it's just not appropriate to carry on. So is your proposal practical financially? Do we have enough money to be able to do what we're proposing to do? Is there a more economical way to achieve this goal? And we need to justify why we're doing this experiment and not that experiment. And my final favorite, have you considered this or that in your approach? Often people will sit there in the audience and they will try and ask you a question about their research. And sometimes this is really helpful. And no, I haven't considered that in my research, but actually that's really relevant and that's a really fantastic point. Other times it's completely irrelevant and they just want to ask a question to sound smart. And it's very difficult because I'm thinking, well, no, of course I haven't considered that because it's completely unrelated. So it can be very tough answering questions at the end of academic conferences as the questions can definitely be a little bit mean. So a very quick activity now, another minute. Thinking of those medical research questions that we thought about a few minutes ago, what barriers might you experience in trying to answer these questions? So some of the barriers that I just explained, are there any barriers that you think you might experience in trying to answer those questions? So 1 minute, and again, I will ask you guys to raise your hand. All right, let's come back together. Does anyone feel like they'd like to put their hand up and share some of the barriers they might experience in answering their research questions? Yeah. Sorry, you're going to have to... Yep, definitely lack of technology. Not only that we haven't potentially developed that technology yet, but also not every university, not every department will have access to certain pieces of equipment. So you may not have the technology you need to get the job done. Fantastic. Any other ones? We'll try and take one more. Yes. That's a really good one. Is the research considered ethical? Ethics barriers. Even if you think and you've heavily been thoughtful and considered whether you think your experiment is ethical, there will be a whole panel of people out there deciding whether or not it is again, and it really is up to them as to whether your research is ethical or not. Fantastic. All right. So we're going to have a little look at this academic case study of my research, and my research title, which believe it or not I have shortened, is Discovering Novel Targets for Antibody Drug Conjugate Targeting in Ovarian Cancer Treatment. So antibody drug conjugates are a type of therapy. So if you imagine that's a therapy, we're treating ovarian cancer, and the antibody needs to bind to something, and that's the novel target. Okay, so we're discovering these novel targets for a specific type of therapy in ovarian cancer. So we're just going to break this down a tiny little bit, and we're going to start by looking at ovarian cancer. So what is ovarian cancer? It is the malignant growth of cells on one or both of the ovaries, which you can see circled in the diagram. And why did we decide to study ovarian cancer in particular? There are many other types of, even gynecological cancers out there, endometrial cancer, cervical cancer. Why ovarian? So it has a very high incidence. It has around 7,400 new cases every year in the UK. For women, it's a very high impact cancer, which is the 6th most common cancer in women. It's right up there with breast cancer. And the mortality rate, this is quite a scary statistic, is very high. 11 women every day in the UK will die from ovarian cancer. And it's coupled with a very poor survival. So survival rate is defined as surviving more than 10 years after you've been diagnosed with your cancer. It usually means after 10 years that with a bit of luck, you're kind of out of the woods. But only 35% of women do survive 10 years after their diagnosis. And we're going to look at point number 5 in a little bit more detail because of its late diagnosis. It's very difficult to detect early. It's much more commonly detected later in life, and it has its highest incidence in women around 75 to 79 years old. So we're going to take a bit of a closer look there. So why is it diagnosed late? Why is it diagnosed at a late stage and late in life? By stage, we stage cancers depending on how aggressive they are, 1 being the lowest and 4 being the highest. Why is it diagnosed late in life and a late stage? So for a start, it has got very ambiguous symptoms. Symptoms like bloating, fatigue, and stomach pain. Those aren't the kind of things, especially people under the age of 30. If you go in to the doctor's surgery and say you're experiencing these symptoms, it is very unlikely that your doctor is going to recommend you going for an ovarian biopsy. In fact, they're probably going to look at you like you're a little bit crazy. You know, you're 30 years old, you're a woman, you're tired, bloated, and you have stomach cramps. What do you think's wrong with you? It's probably not ovarian cancer. So doctors really need to, and they struggle to decide whether or not these symptoms are worth giving somebody a biopsy for. It's very stressful going in there, having a procedure where even though it's keyhole, you still have to have an incision in your stomach to get to the ovary. Do they want to put you through this when really, you know, you're just bloated and a bit tired? Probably not. But by the time the symptoms become alarming enough for us to warrant, okay, there's definitely something wrong here, the reason that the symptoms are, presenting themselves more is because the cancer is already at a late stage. So very early on, the symptoms are so vague, you wouldn't even know that you had it, just to terrify you further on a Thursday afternoon. So really this highlights the fact that we need a high throughput, specific diagnostic test. So this means a type of test that we can do quickly, we can do cheaply, and we can screen many women, as many women as we can over the year, okay? So cervical cancer is a really good example of this. So cervical cancer has got a high throughput diagnostic test. Once you're over the age of 25, you will be offered the chance to come in for a quick smear test every year. And this just simply looks for cancerous cells in the cervix. And it's so quick, very easy, not exactly the most pleasant thing in the world, I imagine, but it's not surgery and it's definitely not cancer. And it's a lot easier for women to be screened in this way. So we need something like this for ovarian cancer. So the current situation today, how do we diagnose ovarian cancer today? Normally it's diagnosed in a routine pelvic exam. You might not even be going in for any kind of nasty symptoms. It might just be routine at perhaps one of these clinics where you go in to have your smear test. Blood biomarkers, so CA125 is one of the markers that we use in the blood that's elevated in ovarian cancer. But the problem with that is that it's also elevated in other conditions and other cancers. So even still, high CA125 isn't enough to warrant somebody under 30 going in for surgery. Okay. Ultrasound, we're looking at kind of clinical imaging and can you visualize a tumor that way? But of course, if you can see it, it's already at a somewhat late stage anyway. So again, that's not particularly helpful with early diagnosis. Surgery and biopsy is the only confirmatory method in which to test for ovarian cancer. And normally, in the same surgery, it's coupled with the treatment. So if you have the surgery to take the biopsy and a mass is found, then you take the mass out within the same surgery treatment. You then follow that up with chemotherapy and radiotherapy, as we talked about before. The problems here being obviously CA125 not being specific. Surgery can sometimes be suboptimal. This is not through the fault of the surgeon. It doesn't mean that they've done a poor job. Sometimes, because especially at the cellular level, you can't see if you're leaving behind one malignant cell. And we know that that one little malignant cell can proliferate and make the tumor regrow back. Or sometimes those cells might be somewhere where you physically can't reach them. Okay? And we all know that there are very distressing side effects to chemotherapy and radiotherapy. So a little bit on antibody drug conjugates. This is the type of therapy that I'm working on. So this is making the drugs that we use in chemotherapy. So this cytotoxic drug here, you can see in red, we're using that a monoclonal antibody to link them together to make this more targeted. So to put it simply, chemotherapy is a whole body treatment, okay? It treats all the cells within the body, and this has a very high rate of toxicity. That's why we get such nasty side effects. If we link that to an antibody that only targets the malignant cells, we can reduce that toxicity massively by only targeting the malignant cells and leaving the rest of our body largely alone, which decreases the side effects and increases the quality of life. So this antibody that is linked to a drug has to bind to a particular protein or target, a particular protein that is present on malignant cells but not present on healthy cells, okay? And it's discovering these targets that form a large part of my research at the moment. So another key characteristic of ADCs, antibody drug conjugates, is they have to internalize into the cell. So this cytotoxic drug that's linked onto there, he's very stable outside of the cell. He's not going to be performing any of his functions. But once they're inside the cell, it breaks down and then it can kill that one cell specifically, reducing overall body toxicity. So what questions am I trying to answer? Is there a good target for ovarian cancer? Is there a good target out there that's present on the surface of malignant ovary cells and not present on healthy ovary cells? What makes a good target? Are they on the membrane and do they internalize into the cell? So novel ADC targets for ovarian cancer, they should be expressed highly in malignant ovary. They should be minimally expressed or not at all expressed in healthy ovary. They should be on the membrane and they should internalize into the cell. So these are four main characteristics of proteins that we're looking for. And based on this criteria, we recruited somebody to develop a mathematical algorithm to search through multiple databases for promising targets like this. And we had a massive list and we narrowed this down to 6. And I'm going to just show you 2 of them. So A1 and A2, you can already see if the yellow is the plasma membrane on the outside of the cell, you can already see that these proteins traverse the membrane like this. They have intracellular regions that are inside the cell and extracellular loops that we can target with antibodies for a particular therapy. And the images on the left-hand side show you that they are more highly expressed in ovarian cancer. So healthy ovary at the top, and the 3 boxes below that, serous, mucinous, and endometrioid. These are all subtypes of ovarian cancer, and the brown stain means that there's more of that target there, okay? So you can see there's lots of blue in the healthy ovary, okay? And then we're getting more and more brown as we move through the different subtypes. So this is already looking like these targets are very promising. So how can we tell for sure? Because this data was obtained in silico using a computer database. How can we make sure about this? So we bought in commercially available cell lines and we did the experiment. Are they on the membrane? That's what we're hoping for. So in blue, you can see the DAPI is staining the nucleus of the cell to make sure that I've actually got cells there. The green is looking, it's a control for the membrane. We're interested in membrane proteins. We need to know that the membrane isn't disrupted or weirded out in any way. And finally, the red is the target, is my protein of interest as to what I'm looking for. So what we did was we have an antibody that targets that protein, and attached to that antibody is a fluorescent dye, and this particular one fluoresces red. So it will go in, it will bind to the target, and we'll be able to see exactly where it is underneath the microscope. microscope. So you can see, thankfully, that these guys have got nice little halos surrounding the cells, which is really nice because it means that it's on the membrane. So, last few slides, nearly there. So this is moving into primary cells. We work with women in the hospital next to us who donate parts of their ovary to us, and we extract primary cells from that, and we check to make sure that it's present on the membrane of those cells as well, moving closer into human models. And yes, we can see that they are present there. So, do they internalize? Okay, this is the big one. It's all well and good if they bind, but if they don't internalize, they're not going to do anything at all. So we use the same process, this antibody going in and attaching to the target that we're interested in. However, instead of a fluorescent dye, this time we attached a pH-sensitive dye that only fluoresces when it's inside the cell due to the change in pH. Okay, it's different inside the cell than it is out. So whenever this protein and the antibody goes into the cell, then this dye will fluoresce. And thankfully, you can see some little red blobs on the diagram behind me. And that means that my protein and the antibody is indeed going into the cell, which is fantastic. So, challenges in my research, antibody validation. How do I know that my antibodies are binding to the target that I'm suggesting they're binding to? Healthy controls, finding healthy women that want to donate their ovaries so that I can have some controls, very challenging. Finally, monoclonal antibodies and time. It is very time-consuming and expensive to produce monoclonal antibodies. Okay, the last couple of slides are just summaries and reflections, so we're just gonna skip over that, and you guys are now free to move on to your next session. Thank you very much.